Hi students, we're going to start looking at some more of the details, some more of the principles that are involved in regulating metabolic pathways. Let's look at the metabolite glucose 6-phosphate. We'll look at it in particular in a liver cell because it has a number of potential fates that it could take once it is phosphorylated inside the liver cell. The glucose could be dephosphorylated, could be returned right back into the bloodstream and be used to maintain adequate store of sugar in the bloodstream. It could also be taken down the pentose phosphate pathway so that we're able to generate nucleotides and build up our stores of NADPH, so that the anabolic pathways will have plenty of energy available to them. Of course, the primary pathway that we would expect it to take is through glycolysis and all the way through the respiratory pathways to generate ATP for the cell. It could just go as far as acetyl-CoA and then be diverted into the generation of fatty acids, sterols of various types so that we have enough cholesterol and we have enough of the steroid-based hormones. Could be converted into other types of sugars for purposes like glycosylating proteins, to give them their functional properties that they need, or the glucose 6-phosphate could become a part of glucosamine. It could be modified into glucosamine or other sugars that end up being a part of the extracellular matrix polysaccharides. So important in various types of connective tissues like cartilage and just a number of different places in the body. Or maybe we want to store it to maintain an adequate stockpile. And so it will be converted to glycogen and then used at a later date as needed. With all these potential fates for even one metabolite, let alone all the metabolites that are in the body, we can readily see that pathway regulation has to be very sensitive, and it is by its very nature complex, especially when you consider all the environmental changes that are just a part of everyday life. Our energy requirements are always changing. Are you exercising? How much are you exercising? What kind of exercising are you doing? And how intense is it? And so on. What is the state of your immune system? Are you trying to heal a wound right now? Are you recovering from some type of an illness? Also, what kinds of nutrients are available and how much? How long has it been since you've eaten? How much fat versus protein versus other type of nutrient was in that last meal that you ate? For any metabolic pathway, regardless of the number of individual metabolites involved, the concentrations of those metabolites will remain constant. However, the flux, the rate of flow through the system, is going to change with availability of substrate, buildup of product, necessity to use alternative pathways instead of this particular one. Those things are always subject to change. Now, the rate of the individual reactions, V1, V2, if we had more steps, V3, V4, all of those rates would be equal to one another in order for the concentrations to remain constant. So this dynamic steady state is a condition where we would say the cell maintains molecular homeostasis. What if the cell type changes? Maybe we're talking about a stem cell in one instance, maybe a hemocytoblast, for example, and then we're talking about its conversion to a mature erythrocyte, a mature of any type of the leukocytes, or even platelet cells. In those cases, the concentrations at which the individual metabolites are maintained may change, but they're going to be set at a new normal, and molecular homeostasis will once again become the dominating condition. In light of the cell's need to maintain this molecular homeostasis in the face of constant changes in environmental factors, constant changes in the needs of the cell, then the enzymes that are responsible for flux through the pathway are going to change in concentration accordingly. So we ask the question, how many active molecules of any given enzyme, we'll call it X, are present in the cell at any given time? What factors contribute to this process? We'll describe these individual steps on the next couple of slides in more detail, but this one gives us an overview of 10 different factors, and it's not exhaustive, things that influence the activity and the total concentration of any given enzyme in a cell according to need. Now what we see is we start out with transcription of messenger RNA. In any cell, all the messenger RNA that's present makes up its transcriptome. That transcriptome, at least a certain subset of the transcriptome, becomes translated into proteins. And so all of the proteins that are available in the cell make up its proteome. Then it's proteins that become active components of the metabolic processes going on in the cell are in total described as the metabolome of the cell. In order to build up a stockpile of enzyme in the cell, we have to, of course, begin with transcription and go on through translation. So initiation of transcription of any given gene in the cell is going to be a response to a specific extracellular signal or a specific set of extracellular signals binding to cellular receptors. We may have the production of transcription factors that bind to the DNA of that particular gene in a region of the promoter called a response element. The binding of transcription factor to response element may serve to enhance transcription, may serve to inhibit transcription, to speed up or slow down the process so that more or fewer transcripts are made. Now that messenger RNA, once it's made, is going to need to go out to the cytoplasm where ribosomes associate with it and begin translating polypeptide chains. How many ribosomes end up translating that particular strand of messenger RNA is variable. How close together the ribosomes can be on the strand, how long the messenger RNA remains viable before it gets degraded. Those are all factors that are set by a number of regulatory mechanisms within the cell. Translation gave us a polypeptide chain that then will be modified into the enzyme. But even as the enzyme, however, it does need to be divvied out or sequestered in the proper subcellular organelle where its job is required. Now, many enzymes are not going to be functional until they're associated with the proper regulatory proteins. They may need cofactors associated with them. There may be ligands that need to bind to the enzyme to allosterically affect its activity. A number of enzymes, as you know, are going to be phosphorylated or dephosphorylated in order to switch their activity on and off or to modify the degree of activity that they have. And then the availability of substrate is important. An enzyme is worthless if there's no substrate available. But once the enzyme is activated and it binds its substrate, then it will be able to generate the product. Eventually, however, though, proteins become less and less efficient at what they do as individual molecules, and so molecules that reach this stage are going to be tagged for degradation. Ubiquitin is a type of a label that gets added onto molecules of protein that have approached their limit of their lifespan, and so those proteins will be sent for degradation at a proteasome after they've been ubiquinated. Different tissue types, as you can see here, produce different kinds of proteins, and these different kinds of proteins have different average half-lives. Some are much more stable. like those contractile elements that are found in muscle tissue or in heart tissue, or even in the neural tissue of the brain and so on, whereas other proteins turn over very rapidly. If we look at the liver, that serves as the opposite extreme, where the half-life of the tissues, the half-life of the proteins is actually less than a single day. Take a look at this reaction pathway. Since the net rate of each step in the pathway is the same, 10, we know that it's in steady state. However, if we look at the rates for the forward and reverse reactions of each individual step, we can see that while reaction number 2 and reaction number 3 are very close to equilibrium, reaction 1 is far from it. So if we make slight changes in the concentration of C or D, that will reverse steps 2 and steps 3, but you just really cannot change concentrations enough to reverse step 1. This is a good table to spend some time studying. If you look at it, all the enzymes in the table are enzymes that we talked about in the previous chapter, and it gives their equilibrium constants and their standard change in free energy, but it also gives the mass action ratio in different types of cells, liver cells and heart cells. So in vivo, then some of these reactions are near equilibrium and some are not. And we can see that the delta G actually in the heart is not always very close to the delta G under steady state conditions. If a cell is going to regulate its metabolism, it is crucially important that the concentration of ATP remain constant all the time and that it remain constant at a high level. This is important both kinetically and thermodynamically. Kinetically, if you look at the graph on the right, you see that as a Michaelis-Menten type equation relationship. And what it shows us is that if we maintain a high concentration of ATP at all times, then those many enzymes in the cell that require ATP in order to function are going to remain fully saturated so that the reactions that they mediate will be able to take place at a high rate. Thermodynamically speaking, it's the ratio of ATP to ADP in the cell that's so important in maintaining a delta G that favors the forward reaction. As long as we maintain a high concentration of ATP relative to ADP, then we will maintain the thermodynamic push toward completion of the reaction. It's interesting to note that really the most important concentration in many cases as far as regulating metabolism is the concentration of AMP. If you recall, we talked earlier this semester about the ATP replenishing reaction that uses adenylyl kinase so that two ADPs can react with one another, producing AMP and another ATP. Thus, the depletion of ATP in the cell is partially modulated by the second reaction, but the production of AMP is going to continue so that the concentration of AMP goes up and up and up as long as adenylyl kinase is able to continue with the reaction, replenishing ATP, but in so doing, building up more and more supply of AMP. When AMP concentrations rise in the cell, whether it's due to depletion of ATP, high levels of exercise, or whatever, it activates a very far-reaching enzyme called AMP-activated kinase. This figure shows us that it has effects at the skeletal muscle, at the heart, and at the brain, increasing certain activities that will allow us to increase our energy stores and at the same time blocking activity at the adipose tissues, at the liver, and at the pancreatic beta cells so that we also conserve the energy we already have in storage.